Hi everyone, my name is Shani Neomi Weitman, and today I'll be presenting my final project for the Nutrition Cardiometabolic Health class as part of the Doctorate in Clinical Nutrition program. My paper explores the role of nutrition, the gut microbiome, and endogenous glucagon-like peptide 1, known as GLP-1, physiology in cardiometabolic disease, and proposes a physiology-driven clinical protocol that's centered around restoring metabolic signaling. GLP-1 receptor agonists have transformed the treatment of obesity and cardiometabolic disease by producing significant weight loss and improving glycemic control. However, evidence showing weight regain after treatment discontinuation raises an important question. If GLP-1 receptor agonists stimulate metabolic signaling pharmacologically, can nutrition and targeted modulation of the gut microbiome help restore the body's own endogenous metabolic signaling? So that question became the foundation of my review and ultimately led to the clinical protocol I proposed. And to answer that question, I first had to understand the underlying biology. One of the strongest themes throughout the literature was that cardiometabolic disease is not driven by a single abnormality, right? Instead, it develops through multiple interconnected physiological disturbances, including visceral adipose tissue dysfunction, chronic low-grade inflammation, insulin resistance, leptin resistance, alterations in the gut microbiome, and impaired endogenous metabolic signaling. These pathways reinforce one another, which helps explain why addressing only one mechanism may not fully resolve long-term metabolic health. Now, this is where the literature became interesting. Rather than directly stimulating the GLP-1 receptor, nutrition works throughout the gut microbiome. Dietary fiber is fermented by the gut bacteria to produce short-chain fatty acids, which activate GPR41 and GPR43 receptors on intestinal L cells and stimulate endogenous GLP-1 secretion. The two microbial species that stood out throughout the literature were Akkermansia, that support gut barrier integrity and insulin sensitivity, while Faecalibacterium produces butyrate and help reduce chronic inflammation. So together, these findings provide the physiological rationale for the clinical protocol I propose. Based on the evidence reviewed, I developed a physiology-driven clinical protocol that addresses the underlying drivers of metabolic dysfunction rather than focusing on body weight alone. It begins with a comprehensive evaluation to identify each patient's metabolic profile, followed by an individualized treatment plan. Nutrition serves as a foundation of the protocol through an anti-inflammatory dietary pattern that eliminates ultra-processed foods while increasing fiber, resistant starches, and polyphenol-rich foods to support endogenous metabolic signaling. The next step focuses on targeted modulations of the gut microbiome by promoting Akkermansia and Faecalibacterium. Finally, patients are monitored over time using both clinical outcomes and metabolic biomarkers so the treatment plan can be adjusted as metabolic health improves. In conclusion, this review reinforced that cardiometabolic disease is driven by multiple interconnected physiological disturbances rather than a single hormonal pathway. The literature suggests that nutrition and the gut microbiome influence many of the same physiological pathways targeted by GLP-1 receptor agonists through different biological mechanisms. Based on these findings, I propose a physiology-driven clinical protocol centered around restoring endogenous metabolic signaling through nutrition and targeted gut microbiome modulation. While this hypothesis requires prospective clinical validation, I believe it provides a strong physiological rationale for future research. Specifically, future studies should investigate whether restoring endogenous metabolic signaling can reduce long-term reliance on GLP-1 receptor agonists while improving sustainable cardiometabolic health. And these are my references, and I thank you again for your time.